So, shall we make a start? Okay. Good morning, everybody, and welcome for the Seren event today. My name is Noha, and I'm presenting the medical sciences session for today. And what I'm going to present is the stem cells, basic concepts, and regenerative medicine. So a little bit about myself. I finished my first degree in pharmaceutical sciences, and then I had my master's degree in pharmaceutical sciences, again in biochemistry. And then I decided, I have done all of these in Egypt, and then I have decided to move here for my PhD, and I'm now finishing my PhD in cancer biology. So I have shifted a little bit from the pharmaceutical sciences to cancer biology. And one of the reasons why it's important to study basic biology, because when you are studying basic biology, you will know the problems, you will know the physiology of the systems, you will know the pathology of the diseases, and then you can start designing therapeutics to target them, and then you can treat people. So without studying the main biology of the systems and the problems which are happening in the diseases, you cannot treat them. I'm working in the European Cancer Stem Cell Research Institute, so we are interested in cancer stem cells, but before I can introduce the concept of cancer stem cells, we need first to know more about the normal stem cells before moving to cancer stem cells, and that's what we are going to focus on today. So the normal stem cells, how they are behaving, what are their characteristics, and why they are important, and why more research is being directed to study them now. So the first thing I want you to do now in pairs or small groups, I just want you to think about what could be the relation between a stem cell and a stem of the tree, and you will find some clues which are going to help you in this photo. So I want you to try and think what could be the relation between stem cell and stem of a tree, what could be the importance or the relation between water and what's happening down there, what is the importance or the significance of having this time arrow here. So I'll give you like 2 minutes to discuss it in small groups, and then we can discuss it together. Is that okay? Okay. Okay, so let's share ideas now, please. Thank you very much for the people I have discussed it with them. And then I want other people to share their ideas Now, so if I say one from that side, and it's, I know I'm unfair because this is a small side, so if you can share your ideas about what would be the relation between a stem cell and the stem of a tree.
Well, stem cells are all the same, like the track until they differentiate, and that's when they branch out.
Yeah.
Into different cells.
So the stem cells are able to give rise to cells which look similar to them. So the stem of the tree is given rise to more of the stem of the tree, which morphologically, or it looks like the stem of the tree, and then it's branching and giving rise to branches, leaves, and flowers, which look different from the stem of the tree. And the stem cells are able to give rise to cells which look like themselves and are able to give rise to cells which look differently from themselves because they are differentiated cells. So this is one thing we can infer from here. So if we say from that side, another thing which we can pick. Yes? Anyone? I'll pick a one then. Okay, so if we say, anyone from here, please, if you can tell me what would be the relation between the time ROE, what's happening with time for the tree? What happens with time for the tree? Any answer?
It grows.
It grows. So with time the tree grows, which means that starting from like one cell or small cells, you are given rise to a full tree here. And the same applies for a stem cell, because the stem cell is the origin of life. So starting from a stem cell in the embryo, you are a full human. So it's given rise to all the different tissues present in your body. So from this part, and I want you to be quick now, I want someone to tell me what's happening here.
The stem cells present before they differentiated, they're exposed to various different hormones where they are in the body, and that dictates which future cell they differentiate into according to the needs of the body.
So this person, what is he doing? He is like irrigating the tree, right? Because the tree cannot grow without water and nutrients, right? The same applies for the stem cells. So the stem cells, they need to be surrounded by a microenvironment, and the microenvironment will have the specific growth factors, and these growth factors will dedicate whether they are going to replicate to give rise to cells which are similar to themselves, or whether they are going to differentiate to give rise to cells which are different from themselves, and to which cells they are going to differentiate. Is that clear? Okay? Any other relations picked from this picture before we move? Anyone, any smart one has picked any other thing before we move? Okay, thank you very much for this. So what's a stem cell? From what we have discussed now, a stem cell is a cell which has the ability to replicate itself, so it's given rise to a cell which looks the same like itself, and at the same time, it can give rise to a cell which is different from itself. It's a differentiated cell. So it's a cell starting from an undifferentiated cell. It can then differentiate and give rise to different cells, type of cells which are present in our body. So it can give rise to differently looking cells and differently functional cells. Like, as you can see here, this is a nerve cell which looks very different from a red blood cell, for example. And their functions are different as well. So the stem cell can replicate and give rise to a cell which is another stem cell, a second stem cell which looks like itself by the process of self-renewal. So it's renewing itself, and it can give rise to cells which are different from themselves and they have functions by the process of differentiation. Is that clear? Okay, so a question for you now: what would happen if the stem cells are not able to self-renew? What do you think is going to happen? Excuse me, can I say it again? Die is it's not going to die. So the original cell is still there, but it's not self-renewing. Yes? Yeah, so we are going to lose the pool of stem cells, and these are the cells which are able to give rise to themselves and to other parts, to other cells as well. So which means that we are going to lose this pool by time if it's not renewing itself. So what happens if the stem cells are unable to give rise to cells which are different from itself? What happens if the stem cell is not differentiating? And this is like all the cells in our body, or most of the cells in our body, unfortunately, they are not able to self-renew. So once the damage happens to themselves, they cannot give rise to another cell looking like themselves, and they cannot differentiate. So once you lose a certain type of cell, like nerve cells, they cannot, not too many cells in there, the normal cells cannot differentiate. It's only the stem cells, which is a rare population, which can then differentiate and give rise to the nerve cell. So what do you think is the importance of the differentiation property of the stem cells? Yeah, so when you have a defective cell or a damaged cell, you need to replace it. And because the terminally differentiated cell like this one, like most of the cells of our body, cannot do it, so you need a stem cell so that it can give rise to this differentiated cell and this can replace the damage. So this will be summarized here. So one stem cell can self-renew, or so can give rise to a cell which looks like itself, or can give rise to a cell which looks different from itself. And the importance of this part, the self-renewal, is to maintain the stem cell pool, and the importance of that part, the differentiation or specialization, that we want to replace the dead or damaged cell throughout our life. Okay? Is that clear? Any questions? Okay. So I have got a question for you then. So choose the correct answer. It's a very easy question. By self-renewal property of stem cell, a similar or a different cell is produced. A similar cell is produced by the self-renewal property. So when would be the case when a different cell is produced? What would be the property of the stem cells then? Differentiation. So by differentiation, stem cell will give rise to a cell which is morphologically and functionally different from it. Okay. Let's then move to the types of stem cells. And again, I'm talking about the good normal stem cells. I'm not talking about the evil bad one, the cancer stem cells. For the types of normal stem cells, we have got 3 types of stem cells. These are embryonic stem cells, adult or tissue stem cells, and induced pluripotent stem cells. I just want you to have a think for a minute about what would these names imply about the characteristics of stem cells. Can you guess, for example, from where they are produced, or from where we can separate them, whether they are normally present or they are produced in the lab? So have a think from the names and see what these names would infer about the characteristics of stem cells. Okay, so my first question was, from which stage, in our life are these cells separated? So can you make a guess from names? Yeah, so which one is separated from an embryo? So the source of embryonic stem cells will be an embryo. How about the source of adult or tissue stem cells? So what would the name imply? That they are separated from? From adult. So they are separated from adult or from the tissues from an adult body. Okay. For the induced pluripotent stem cells, what do you think the name is implying as well? So is it normally separated from the body, whether it's an embryo or an adult like the first two ones? Yes? Yeah? Yes, please.
I think we know stem cells are like naturally occurring, they weren't like made to be stem cells.
Yeah, so they are not natural, thank you. They are not naturally occurring stem cells. We have done something to these cells to make them stem cells, and that's why they have got the induced part in the name. This means that this is not natural. We have done something to them in the lab to make them behave like a stem cell. Okay? Okay. So the first type of it, and as you have told me, the embryonic stem cells, where we can find them, so we find them in a specific stage of the embryo, which is the blastocyst, and this is a stage where the embryo is only formed of about 50 to 150 cells. Only these cells have this ability, so we name them pluripotent. The name of pluripotent, so the potency is a measure of their ability to differentiate, and these embryonic stem cells are pluripotent, which means that they can differentiate to any cell type present in the body. And that's how a whole human body is formed from only very small, limited number of cells. Because these cells are very potent, and we name them pluripotent, that they are able to produce all the cells and differentiate to all cells present in our body, which looks different and have different functions as well. So what do you think would be, so this looks like very interesting if we can use them, if we can manipulate these cells, which means that we can regenerate any defective organ in our body. But what would you think would be the main problem with using this type of stem cells? Excuse me? Ethical issues, yeah, that's correct. Why there would be ethical issues concerning this type? Yeah, because you are taking them from an embryo, so you are killing a potential life. And we'll talk about this debate later on. Okay, so for the second type, it's the adult tissue stem cells, and these are stem cells which are found only in as limited numbers present in our tissues. So they are very rare cells present in the tissues, and they have the ability to differentiate to all the cell types which are present in that tissue, but not to other cells which are not in that tissue. What does this mean? So for example, an example here is the hematopoietic stem cells, which are the blood stem cells. Where we can find them and when we can separate them, they are found here in the bone marrow. So if we can take these cells and manipulate them, or even inside of our body, they are able to give rise to all the cells here, which are the cells present in our blood. So to the white blood cells, red blood cells, platelets, all different types of cells present in our blood come from the hematopoietic stem cells. But hematopoietic stem cells cannot give rise to a nerve cell. They cannot give rise to a liver cell. They cannot give rise to a heart cell, because they have limited ability of differentiation. So they are able to differentiate to all the cell types present in that tissue, but they cannot differentiate to cell types which are present outside of that tissue, and that's why we name them multipotent. So they still have got the ability to differentiate, but because the ability of differentiation is limited compared to the embryonic stem cells, they are called multipotent. But the powerful one, which can differentiate to any cell present in our body, we call them pluripotent. Is that clear? Any questions? Okay, so let's now move to the induced one. So the one which we did in the lab, and it has got, the name has got induced and the pluripotent stem cells. So we were able to imply first from induced that they are not naturally present, but that we have done something to these cells to make them a stem cell. So what would we imply from the word pluripotent? Would they resemble the embryonic stem cells or would they resemble the adult stem cells? They are resembling the embryonic stem cell. So in a way, which I'm going to discuss now, we were able to transfer a normal cell, which is a terminally differentiated cell. It's a cell which is unable to self-renew itself or even differentiate. And we have done something in the lab for this cell to make it revert back to a stem cell which is able to self-renew and differentiate. So what we have done in the lab, so it's not we, so it's Professor Yamanaka from Japan, and he won the Nobel Prize for doing this in 2012. And what he have done was reprogramming of the cells. So he started from like a pool of 24 genes, which he knew that are important for the stem cell functioning. And he started to introduce them to the cells, and he found that by introducing this whole of genes, the cells have got the ability to revert back to being stem cells, and they are able to self-renew and differentiate. And then what he did next is that he started to lessen the number of genes and try to find the best combination with the least number of genes to be introduced to the cells so that they can be stem cells. And he found at the end of the day, before he won the prize, he reached a cocktail of 4 genes. By introducing these 4 genes into cells, so terminally differentiated cells, which are any cell from your body, like a skin cell. So by isolating skin cells from your body, introducing this cocktail of genes, these 4 genes, these cells were able to revert back to behave like embryonic stem cells. Because we have done this in the lab, they were named induced. Because they behave like embryonic, they were named the pluripotent, because they can differentiate to any type of cell in your body. So the induced pluripotent stem cells, we take a cell, a normal cell from your body, and then by genetic reprogramming, by adding these 4 genes, the cell is reverting back to behave like an embryonic stem cell, and it can give rise and differentiate to any type of cell in your body. So it's a pluripotent, and we have got a huge advantage here compared to the embryonic stem cells, that we are no more needing an embryo. We can take cells from our own body, manipulate them in the lab, revert them back to stem cells, and then we can use them if needed. Okay? Is that clear? Okay, great. So what we are going to do now is that we are going to play. So in groups of 4 or 5, so when you are like sitting like close to each other here, I will give you this task, this jumble task, and the task is like this table, and then we have what should be inside in the wrong place. So it's not placed correctly. What I want you to do, so I'll give you a table which is plain, and I give you these pieces which are cut, and I want you to place every piece in the correct place. I'll give you like 5 minutes to do this, and then I start to pick up, if you are not raising your hands, from like every 2 groups to ask to fill one of these places correctly. Is that clear? Is the game clear? Okay. Let's play then. confidence. Yeah, so I'll get an answer from you then, okay? We will start now with this first row. So the row with pluripotent stem cells, like embryonic stem cells, induced pluripotent stem cells, and ignore about the somatic cell nuclear transfer cells. But these are the pluripotent, the embryonic and induced, both of them together. So where do they come from? Here, this group. Yeah, so yes, so they come from human blastocysts, yes, and then the iPS cells are derived from reprogrammed somatic cells such as the skin, right? Do you agree? Do you agree? Any other answers? Do you think that they are correct or not? Yeah. Okay. The second one is the cells they are able to make. An answer from here.
Any cell in the body.
So the cells they are able to make is any cell in the body. Do you agree with this answer? Yes. Okay. Advantages of these stem cells. I want an answer from here. Yes, from this group. So what are the advantages of these stem cells? Yes. What are the advantages of these cells? Yes? Yeah? And can be manipulated to grow into different cell types in the lab. This one. Do you agree with this? Yes. Okay, the last one, the drawbacks of these stem cells. I want it from here.
Due to the pluripotent nature, they also carry over to cancer.
Yes. Okay, this was the answer? Yeah. Okay, great. Thank you. So for the other one, so the second row is the multipotent tissue stem cells, like the cord blood cells, the other stem cells such as skin stem cells and muscle stem cells. Where do they come from? From here.
Undifferentiated cells and tissue.
Okay, so again, so the following, thank you. So the following one is from here. Do you agree with this? Yeah. You agree with this answer? So from here now. The cells they are able to make. The cells they are able to make. Excuse me? So usually only the type of cells or tissue that it's derived from. That's right. Do you agree? Yes. Okay. Then the advantages of these stem cells from here. Anything. They are capable of limited self-renewal, but would this be advantage? Let me check. Limited self-renewal. Rejection. Yeah, so it's no tissue rejection and cells are derived from the patient. So there is one important advantage here, that there is no tissue rejection if the cells are derived from the patient. This is very important, that there will be no tissue rejections. Okay? And the last one, which should be the easiest, the drawbacks of these stem cells. What are the drawbacks of these stem cells? Yes, thank you. So this is the main disadvantage. Okay? Is that clear? Any questions? Okay. So let's move now to the clinical application of stem cells. So I said that stem cells are now, there are two main research which is being directed towards stem cell research, and too much money is being pumped in this sort of research. So why the stem cell research is appealing and why we think it's important? So because of three uses, and the first one I think is the most important use of it, is the use of stem cells in regenerative medicine. It can be used in drug testing and in disease modeling. So what do we mean by regenerative medicine? So if you have got like a defective organ or something like this, what we tend to do if you have got a defective non-functioning liver is that you will have a liver transplant from someone who should be like matching with you, and then we are having the problems with rejections and other many problems as well. So the hope that with using the stem cell technology that we can generate the organ in the lab, for example, or start generating with small amount of cells and then introducing them into your body, and then the damage can be replaced by a newly functioning organ in your body. We will talk a little bit more about regenerative medicine in moments. So how can we use it for like drug testing and disease modeling? For drug testing, for example, we have got this bit of the photo. So it's identifying drug targets and test potential therapeutics and toxicity testing. So if we can model a part of the body in the lab, so if we are having a part of a liver tissue or a heart tissue, for example, in the lab, we can start then putting or introducing our drugs and therapeutics to it and see whether they are toxic or not. So for toxicity studies, we want to see if you have got a drug which is strong to target a certain disease, we want to make sure that this drug is not badly affecting the other systems or tissues in your body as well. So it can be used like this to generate other tissues and test whether the drugs are toxic to these cells. And it can also use to identify drug targets and test potential therapeutics. As I said before, it's very important to identify the normal physiology of an organ or a tissue, to identify what goes wrong when there is a disease. By identifying these targets, you can then try to design a drug which can then target this defect. And after designing it, you can test it in sort of bodies. Because what happened now is that we are testing it, so we test it like in small cells, and then we move to test it in animals, and then if they look promising in lab animals, we move to test them on the humans. So maybe this would be more of resembling to the human, so it could accelerate the process of drug approval. So that's one of the promising uses of stem cells as well in that sense. The last thing is this disease modeling. So as I said, if we know from the biology, for studying disease biology, that the defects in these diseases are caused, for example, by defects in certain genes. So if we can introduce these genes into the cells, so if we can reprogram the cells to have this model of the disease, we can then have a small model of the disease to study more its functions and physiology, and to study the interaction of different genes, for example, in that disease, and then we can target this disease as well. Okay? Is that clear? Any questions? Okay. So let's move to the direct and differentiation. As I said, so to have regenerative, to be able to use stem cells in regenerative medicine, so we will need to direct the differentiation of the pluripotent stem cells to give rise to any cell in the body. And to do this, so some of the companies are now being established, especially for producing reagents which can direct differentiation, so that we can study in lab whether or not we can direct differentiation. And directing differentiation depends on, as we have seen in the first photo in that slideshow, it depends on the growth factors or the signals which are surrounding the stem cells. So what's happening now is that we are trying to tailor different media which we are putting on the cells. So when you know that, for example, to get a nerve cell, you will put these different combinations as growth factors in the media, and if you want a cardiac cell, you will put other ones. And I will only give you 2 or 3 examples from this very complicated photo. So we are starting here by pluripotent stem cell. Either it's embryonic stem cell or induced pluripotent stem cell. So if we wanted to end, for example, by let's take this neural crest stem cell, you will have to take these cells and add, if you can see, so you will be adding factors from here. So you will add different factors to reach this stage, and then you are going to change the factors in the media to reach this stage, and then you are going to put more specific factors which are going to change it from the neural progenitor cell to the neuroplast stem cell. And then whenever you want to go down to more differentiated cells, you are going to change the medium. So the factors in the medium and the factors you are putting, which is similar to your own body, will differentiate the cell, this cell, to the cell you want to do. So this is, again, this is research which is accelerating very much now, so that we can know, we can try to understand what other factor combination, on which plates we can grow these ones, because again, the type of the plate would affect the growth. So what would be the best combo in that sense for all of these things to differentiate this stem cell into a liver cell, into a heart cell, into a nerve cell, and then we can use them. So how can we use them? These are examples of the clinical trials which are ongoing now. So we are trying to use stem cells to treat a certain type of eye disease, this one, to treat a certain type of nerve disease, the Parkinson's disease, spinal cord injuries. We are trying, so there are some trials trying to use stem cells to treat diabetes as well, myocardial infarction. I had a small group which I was tutoring in the last term, and their project was on giving me examples of the use of stem cells in regenerative medicine, and I had fantastic articles written by them. Every one of them went and chose a disease, and they showed me how the stem cells, the research is being accelerated. So there are many more examples other than this, but these are the ones which are now in the phase of clinical trials. So there is a promise in these ones to be treating the diseases in them. Okay, so the stem cell debate. I think I may be finishing with this. So what is this comic trying to say? So we are having two persons here who died. The first one, like a grandpa or someone like him, he said, I died waiting for embryonic stem cell research to find a cure. And he's asking the other one, what about you? So the other one appeared to be the embryo. He said, I was the embryo. So what do you think, as you have mentioned before, the main thing about the use of embryonic stem cell are ethical issues, because you may be killing a potential life. But do you think that's only embryonic stem cells that carries ethical issues and debate with using them? Or would this be reflected on all types of stem cells as well? Yeah, you can share your ideas, please. Yeah, so once we use like induced pluripotent stem cells, we should be happy and there shouldn't be a problem, right? Unfortunately, this is not the case. We are still having debate about using pluripotent stem cells generally, and we are having, because there are knowledge gaps, we don't know everything about them. We have got disagreements on uncertainties, and we have got questions concerning like the patient safety. So are they safe to be used for patients? We don't have enough data to make sure that they are safe, going to cause other problems in the patients. The treatment efficacy, so whether after we are doing all this research, would this treatment be really the magical treatment for this disease or not? There are still some uncertainties here. The suitability of these cells for drug testing and disease studies would be like the ideal model really, if they are using. And social question like the accessibility to treatment, whether governments should really be pushing money and pumping money in this research, or would it be better for this money to be used for other things which are going to affect the society better, in a better way? So these are all sorts of questions which are still making this sort of treatment under debate till now. Okay? I hope you have enjoyed the session. Unfortunately, we don't have time to cover cancer. Thank you very much. Thank you.